אוקיי, נחזור עכשיו להצגות של מלגאים. נעבור למחזור הטרי יותר. נתחיל במתן אילת, מתמטיקה מכון ויצמן. מתן, בבקשה.
Okay, so my name is Matan from Weizmann Institute in Math Department. My advisor is Professor Boaz Klartag, and my field is called Riemannian geometry, metric geometry, something in between. So I'll try to give a brief introduction to what the main object that we study. So generally there are two points of view on what a Riemannian manifold is. So one of them is called the extrinsic point of view, where it's just a subset of the space. So you can imagine, okay, I didn't have time to inflate the globe, but so the simplest maybe examples are just the Euclidean plane, just the flat floor or wall or whatever. And the second is the sphere. So it's just a subset of the Euclidean space that admits a local parameterization. So in the globe, you have longitude and latitude, so you can parameterize it by two variables. So it's a two-dimensional Riemannian manifold. So that's one point of view. Another point of view is called the intrinsic point of view. So there is no ambient space. It's just some space with some structure that can measure lengths and angles. Okay, these are 2 points of view which are equivalent. Okay, this is called the Nash embedding theorem by John Nash. You might know him from the movie, don't remember its name. So you can imagine an astronaut floating in space. If he looks at the Earth from far away, he has an extrinsic point of view. You can see it inside ambient space, but for us or for an ant walking on the surface, we don't see the ambient space. We have an intrinsic point of view. Okay, so these are 2 points of view, but for this presentation, you can always imagine a two-dimensional, some kind of blanket inside space. Okay, so it's a two-dimensional, I will call it a surface, a two-dimensional Riemannian manifold inside space. Okay, so this, when I refer, this is what you can imagine in your mind. And the important thing is that we can measure distances in any Riemannian manifold. So, for example, if I have 2 points in the Euclidean space, the distance is just the length of the line connecting them, right? We know. And on the sphere, it's just the length of the great circle connecting them. Okay, so for example, if I have the unit sphere, and I'm supposed to have a model, but imagine the unit sphere, and I have 2 antipodal points, opposite points, so the distance is just pi. Okay, it's just half the parameter. Okay, so this is just a brief introduction of what a manifold is. So my research in particular is regarding something called isometric embedding. Okay, so I'm not going to get into definitions, but what's the main idea? The main question is, what can we learn about this surface? And we are given an arbitrary surface. Okay, again, some kind of blanket or whatever, hills and valleys inside the space, and we know several distances between pairs of points. Like I said, any 2 points have a distance between them, and we are given this data of distances. What can we learn about the manifold? Okay, this is the main objective. So let's give an example. So imagine the flat Euclidean plane. If I want to find, let's say, 3 points that have distance 1, each pair have distance 1 between them. Okay, so this is just simple. I can have the equilateral triangle, the משולש וצלעות, right? And every 2 points will have the same distance, let's say 1. Okay, but what if I want to find 4 points? Can you think about 4 points in the plane such that any 2 of them have distance, the same distance? Maybe. No, so in that case you have the diagonal that doesn't have the same distance. Okay, so there are no 4 points. I will ruin there are no 4 points in the Euclidean plane that have, every 2 of them have distance 1. Okay, however, on the sphere, for example, you can find 4 such points. Okay, you can put 1 here and 3 of them somewhere around here, and you can find such points. So this example, what I wanted to express is the idea that, for example, if I am given some manifold, and I tell you, okay, on this manifold, I have 4 points that have the same distance, then I know for a fact that it's not the Euclidean plane, right? I can rule out this option. So I only had partial data on my points, but I was able to deduce something on my entire manifold. Okay, maybe I'm the sphere, maybe I'm something else, but I can rule out this fact. So this is the kind of phenomena, the kind of things that people do in isometric embeddings, one of them. And my specific, so this is joint work with my advisor, Boaz Klartag, like I said. So this is the main result. So again, imagine the Euclidean plane, and imagine the points with integer coordinates. Okay, so you have the origin, 0, 0, 0, 1, 1, 0, 1, 1, all the integer. And you have between any two of them, you have the Euclidean distance. So between 0, 0 and 0, 1, it's 1. 0, 0 and 1, 1, it's square root of 2. Right? Any two of them have distances between them. And suppose that now you are given an arbitrary manifold. And on this manifold, I was able to find points corresponding to these integer points that have the same distances. Okay? The point corresponding to 0, 0 and the point corresponding to 0, 1 will have the distance on the manifold that a priori might be anything, have the same distance as in the Euclidean plane. So the result is that this manifold, a priori arbitrary, must be the flat plane. It can't have any hills or valleys. It kind of, you can imagine flattening the fact that I have these points, flattening the manifold. Okay, another way to look at it is if I take I start with my Euclidean plane and I make a tiny bump in the Euclidean plane, some integer points will not be in the same, won't have the same distance. I must change some distance between points. Okay, this is a rigidity result. It's called a rigidity result. And philosophically, we could think about it like the geometry of the Euclidean plane is somewhat embedded inside this relatively sparse set inside. Okay, it's a discrete set. The points can be, doesn't have to be of distance 1 from 1 another. It can be 1,000, right? It's just the simplest example. But relatively sparse set, knowing these distances between them determines the entire geometry. Okay, so the geometry is somehow of the Euclidean plane, and we hope of other surfaces as well. We don't know yet. This is the future. Things we hope to prove. So it somehow encodes the geometry of the Euclidean plane. Okay, so this is the main result, and if you have any more questions, then.
Okay, thank you. Questions? Okay.
So, can you sketch very briefly how you go about proving such a?
Okay, I can tell you the main. Okay, so the main thing we investigate is. Okay, so we imagine having on our surface some kind of points that we know the distances between them. Okay, so we connect them using geodesics. Okay, geodesics is you can think about a length minimizing curve. So we connect them, and at each of these points, we take sequences going to infinity in some directions, and that way we define some kind of direction, like a direction at each of these points, and we use this and Lipschitz functions. It's hard for me to I can tell you later. I can tell you more about it later, but mainly it's investigating geodesics, going through these lattice points or net points, or this is the main ideas.
Just as a reminder, we hope that we are leaving the refreshments here, and for those who spoke in this session and didn't have a chance to get involved in the discussion, this is the time for you to get them, just like this example. Hopefully, you'll stay behind if you have something that you want to discuss. Yes, please.
I understand that on the sphere you said that it's possible to find 4 points of equal distances.
Mm-hmm.
Suppose you increase the number of points, let's say to 5 or more. Do you know what happens
It's
there?
2 dimensions below. It's always, if you want 5 points, you need to have the sphere, 3-dimensional sphere if you want the. Yeah, you have a very explicit characterization of which metric spaces finite metric spaces. You can embed inside Euclidean space and inside sphere and inside hyperbolic space. You have explicit.
So I need to increase the number of the dimensions of the sphere in order to find more points of equal distances.
Yeah, so if you think about it, how do you find the equal distances inside the Euclidean plane? You just take the regular simplex. Right? So a triangle is it's a simplex with 3 points, and if you add another point and you make a regular simplex, it's 4 points equidistant, right? And you can embed it inside R3, right? So if you take the sphere around the centroid and you inflate it enough, they will all be on this sphere. So this is the way.
So hi, I'm Nitzan. I'm from Victor Eldar's lab at Aviv University, and I study interactions between bacteria and the viruses that infect them. These are called phages, and you already heard a bit about phages earlier from Noam. He presented a great research, so I hope it will help everything I say be clear. I'm going to talk about work I'm actually working on right now, and I try to make it as accessible as possible, and I hope biologists won't be bored. Yes. Oh. Okay. So just a reminder, what is a virus? Sometimes viruses are called just bad news in an envelope. The bad news in this case is the genetic material. That's the squiggly line there. And the envelope is this protein structure. And this is a useful metaphor, bad news in an envelope, because in this state, the virus is inert. It really can't do anything. But how does it reproduce? Well, when it encounters a cell, so in this case, this is the bacterial cell, and the virus is called a phage, it can inject its genome into the bacteria, and then the cell can't help but follow the instructions that are written in this genetic code. And in this case, the instructions tell it to make more viruses. And more viruses are made, and eventually they burst out of the cell, they kill it, and they search for other bacteria to infect. But bacteria don't just wait there to be infected like sitting ducks. They have defense mechanisms against phages. So this is like their immune system, like we have an immune system. And one mechanism is, for instance, to cut the phage DNA as it enters the cell. And just as an aside, I want to say that part of the interest in studying these defense systems of bacteria is that many of the tools that we have for genetic engineering are based on these systems, because they have the ability to cut DNA at specific points, which is very useful for engineering. But I'm interested in these systems in their natural context. Really, in the last couple of years, there's really been an explosion in newly found defense systems of bacteria against phages, largely led actually by Rotem Sorek's group from Weizmann here in Israel. And for every new defense system discovered, one of the basic things that's done is to characterize its protection range, to see the diversity of phages that it can protect against. So I want to give you some examples. So these are two systems called Disarm and Forest. And Disarm here is shown to have a pretty broad protection range, defending against a wide diversity of phages, while Forest seems to have a much more modest range of protection. And when you see this type of results, which have now become ubiquitous in the field, I think an immediate question which comes up is, what is it about the mechanism of the different system that can account for this difference in protection range? And this is being addressed very widely by many people, but we wanted to ask a slightly different question. We wondered if there are any mechanism-independent factors that bacteria can control to influence protection range. If you remember Efrat and Guy's talk, genes need to be expressed, and they can be expressed to different levels. And this includes the genes for these defense systems. And what we hypothesized for some basic biological considerations is that simply by increasing the level of expression of a defense system, we'll actually get a wider protection range in terms of the diversity of phages protected against. And this is what we see, for example, for a defense system called SPBK. I now drew it as a shield. So under native expression, what the bacteria, the level that it expresses naturally, can defend only against a single phage from this diversity, while when we overexpress it artificially, it has a much more impressive range of protection. So on the one hand, this is maybe kind of intuitive, but on the other hand, I want to say that this is a quantitative change, and it leads to some qualitative difference. You get protection against completely new phages. But now a question becomes, well, why doesn't this happen naturally? You know, why was it so easy for us to give bacteria with this added benefit? And in biology in general, it makes sense to ask these why questions because of evolution by natural selection. So why didn't this evolve naturally? If again we imagine the defense systems as scissors, there are several possible explanations for why this doesn't happen. But one of them is that in addition to the benefit gained by higher expression, you might gain also a very heavy cost. It's also written there, but I wonder if you can guess what type of cost there will be here for increased expression. You are able to guess it, I think. Energetic cost, so that's a good option because it takes, it does.
You limit bacterial diversity because you might start targeting the bacterial DNA.
Okay, so energetics is a good option, but what's actually happening here is autoimmunity. There is a cost of energetics, but it's minuscule compared to the cost of autoimmunity. When you have a lot of these scissors, then you do get a wider range of protection, but also sometimes you attack yourself by mistake. And this is like an autoimmune disease, like we have it in our body. And so I want to say, to summarize, increasing the expression of the defense system holds like a trade-off. On the one hand, you get a wider protection range. On the other hand, you suffer a cost of autoimmunity. And this trade-off between efficient immunity on the one hand and reducing autoimmunity on the other is really fundamental to immune systems in general. Here we find a nice example for bacteria, but also in humans, it's known that some mutations that can enhance immunity are also associated with a higher susceptibility to autoimmune disease. And finally, I want to go back to regulation and say there's several implications for what I showed you, but one implication is that regulation of expression is very important because you can not only control the level, you can also control the timing of expression. So you really want high expression when you're likely to be infected, and you really don't want high expression when you're unlikely to be infected. And there are some cool examples of regulation in nature. For instance, some defense systems are regulated by density. When bacteria sense that they're in a crowded place with other bacteria, they overexpress these systems. And this makes sense. This is like putting on a mask when you're in a crowded place, when you're more likely to be infected and also more likely to infect. But we imagine there will be much more discoveries in terms of regulation of these systems. I didn't put a thank you slide, but I want to thank my lab, and thank you for listening.
Actually, I have a question. When you look at bacteria in phage communities in a natural environment where there's been selection over time, you can envisage two alternative scenarios. What regarding the phages that are present and the bacterial defense mechanisms. So in one scenario, you would say you would see a mutually exclusive situation. So bacteria that have phages that are protected against by the existing bacterial immunity will not have a chance to replicate, and they will be wiped out. So in fact, what you will see is that, right, intuitively, you would expect that you see phages that are there and bacteria that don't have the defense mechanism. But on the other hand, if you think about kind of evolution, why would the bacteria keep, energetically or whatever, keep a defense mechanism against the phage that is not there, right? So we'd expect that, in fact, you see a matching, that the bacteria in a community will have the defense mechanism that is appropriate for the phages that are in their vicinity. Question is, which one is more correct, or is there some kind of steady state equilibrium that is the right solution for this? Has anybody looked at that?
Yeah, that's very interesting. There's works on this, for instance, in our gut bacteria and the phages there. I want to say that generally, what we see is that there's very a high heterogeneity within a bacterial community of what systems different bacteria that are similar or otherwise have. So in a given community, phages can infect some bacteria but not others. And when you look at an evolutionary timescale, you see that defense systems are often gained and lost during evolution. And one of the explanations is they're gained, they eradicate phage, and then they are now just a burden, then they are lost back again, or kept only in a very small population of the bacteria. So both the dynamics and the steady state is interesting. People have looked at this, but I think it's still an open question.
How did you come to the conclusion that it's not energy but autoimmunity?
Yeah, so there are several ways to do this. One way is by something that's actually, I'll tell you a way that's better than what we did, and I'll tell you actually what we did. The best way to do this is to mutate the specific residues which are important for the immune mechanism, but don't affect the energetic cost of making the protein. So you can change one amino acid, the protein is made out of amino acids, from one to another. And then if you still see a cost, it means just energetics, but if once you abrogate immunity, you also abrogate the cost, this is it's highly suggestive of autoimmunity. What we actually did was we expressed a different protein to much higher levels than what we express these defense mechanisms, and show that it doesn't have a cost. So when we take just a random protein and it obviously has an energetic cost to make it, and we see no effect, so we conclude that it's probably due to the defense system activity here, which means autoimmunity.
The cells can afford to spend more energy, that's what you're saying?
Yes, they can
They are not
definitely.
at the limit of energy.
No, they definitely can. There is a cost, but it's nearly undetectable using our instruments.
Yes. You showed examples of two different bacteria that had it's an illustration, but still one with very low defense capabilities and one with really high. So what would preserve the lowly bacteria in surviving? So why wouldn't it go extinct? Is it just because it's not meeting any of these phages, so it's distant from them?
So the other illustration was more to compare two different defense systems, but there is, when we look at bacteria, there is definitely bacteria with a lot of defense systems and bacteria with no defense systems. And in general, there is an association that these bacteria that don't have defense system are really less likely to encounter phages. For instance, if they're intracellular pathogens, their likelihood of encountering a phage within a cell, let's say, of humans is much lower than some bacteria in the soil, for example. And they do have less defense systems, yeah.
Okay. Thank you very much.
Okay, so hi, my name is Amir. I'm a PT student in physics from Tel Aviv. My advisor is Professor Moshe Goldstein, who is actually also an alumnus, so it's really nice to be here. And in these next 10 minutes, I'm going to give you a very brief introduction to my topic of research. I will barely get into my actual research. I'll just try to give you an overview of the field. And so I'm doing theory in condensed matter physics. And condensed matter physics deals with systems that have a lot of particles. And by a lot, I mean like 10 to the number 2 to 23, which is Avogadro's number. And the question you usually ask yourself in these kinds of systems is, well, these particles have some sort of microscopic dynamics. There are forces between the very small particles, electrons, molecules, whatever. But at the end of the day, we see microscopic behavior. We measure values in an experiment or see phenomena in our day-to-day lives. And the basic question of condensed matter physics is how do the microscopic dynamics lead to the macroscopic behavior? And this is obviously a very general question. It relates to a whole lot of topics, different topics. It relates to chemistry, to biology, electronic systems. And to be concrete, let's focus on the latter and think about the simplest electronic system you can think of. We have some piece of metal, right? This thing. And we want to understand what is the resistance of this piece of metal. Okay, so it's a very simple question. We can answer this question easily in an experiment, right? What we can do is simply connect this piece of metal to a battery, right? So we apply voltage, then we have current. And from the ratio between the voltage and the current, we can get the resistance, right? This is what is called Ohm's law. And so we can do this in the lab easily. But what I'm asking is whether we could tell what is the resistance of this piece of metal without doing this experiment, but by looking into the metal, right? So we can look inside the metal, and when we look inside the metal, we see that it is composed of a lot of particles, a lot of electrons, atoms that move around. There are interactions between them, interactions between the electrons, between the atoms, between the electrons and the atoms. It is a very complex system, right? So we want to somehow understand how can we get from this to this. So this is a difficult question because we have, as I said, a lot of particles, and solving a system with that many particles is just not feasible. It's just not something we can do, definitely not on a piece of paper, but also not on a computer. And this is especially difficult, actually exponentially harder if we think about quantum effects without going into details. So because this is difficult, what physicists usually do, they think of effective models, right? So a model is essentially a simplified description of our microscopic system in a way that really reduces the number of degrees of freedom, the complexity, to the minimal number of components that will hopefully capture our effects, right? The effects that we want to see. And so a very prominent modeling in condensed matter physics, in condensed matter physics of solids at least, is thinking of these electrons and atoms as a system, as a lattice, essentially, a rectangular or any other type of lattice, where you have some electrons that can hop around, there are interactions between the electrons, but the key point is that this model is significantly simpler than this microscopic model. And because it is simpler, perhaps it is possible to solve it. It is still difficult to solve it because there are many particles, but it at least gives us hope that we can do something. Right? So physicists think of ways to solve these kind of models with all sorts of fancy methods and approximations, and this is a whole field of research. So okay, so we have our model, and we want to understand if it is any good. Right, so what we can do, we get a prediction from a model, we somehow solve this model, we get a prediction, and then we go to the lab and ask whether the prediction is equal to the measurement. Right, so let's say this is not the case. Let's say we measured something that does not agree with our prediction. So what could be the reason? I'll give you three possibilities. The first is that our model is wrong. Right, so we started from some microscopic system, we tried to dress it down to the bare essentials, but we missed something. We threw away too many degrees of freedom. We simplified too much, and our model just does not describe the system itself, right? So this is one possibility. The other possibility is that our model is correct, but as I said, the solution is difficult, so we could be wrong in our solution, right? So perhaps we tried to solve, but we got a wrong solution, so we got a wrong prediction. But there's also a third reason, possible reason, which is that both our model and solution are correct, but in the experiment, there was something we did not take into account in our model, which caused this discrepancy, such as noise or disturbances or something that was present in the experiment but was not present in the model. And you could say that, well, if we do not include noise in our model, then it is not worth much because there is always going to be noise. But assume that we are in a very basic step, the very first steps of our research, so we do not want to concern ourselves with such intricacies, right? We want to understand the bare essentials. So as a first step, we usually think about the simplest model, which will not include these disturbances. And the key point is that if this happened, if our prediction is not the measurement, we cannot know which of these possibilities happened. Okay, so it is really impossible to know what led to this discrepancy. So what people started doing in the past 15 to 20 years is build what is called simulators, or specifically quantum simulators when it relates to quantum systems. And the idea is very simple. The idea is that, well, we have a model, right? Our system is not the model. Our model hopefully describes the system, but the system is not the model. So what we can try to do is actually take our model and implement it in a lab. Because this is kind of reversing the order, right? So the idea is that you take this simplified model and try to implement it as is in a lab. So for example, in this example, we had electrons that move around in a lattice. We can try to simulate this model by taking something that is not electrons. It will play the role of electrons, but it will not be electrons. It will be something else, something synthetic that will behave like electrons in our model that will be able to move around in some square lattice. There will be some interactions. And this will be a simulation of our simplified model. So we get two things out of this. First of all, we get a clean environment. We have a much better control over noise, over disturbances. We can clean away stuff that we do not want. And also, we get a very good controllability. Because we implement this model, and for example, we can implement the interaction between these electrons, we can also control the strength of the interaction, right? So we can tune the parameters of our model. Essentially, we have a black box with knobs which we can tune in order to really understand how our model behaves. Okay, so this is the essential advantage of quantum simulators. And this is really nice because it really gives you a very nice playground that you can understand your model with. And this is interesting both to theorists and experimentalists because there is a really nice interplay between these two environments, or two species. To understand why that, let's look at the whole flow chart. So assume we have some phenomena that we want to understand, but as I said, the microscopic model is way too complex. We cannot dream to solve it. So we try to think of a model, but the model is still difficult to solve, right? So we simulate it. And from the simulation, we can understand, for example, if our solution was correct or wrong, whether we can or we should improve our model, how can we include noise and describe noise. And essentially what we get is insights about the original model we started from. Okay, so that is the basic concept of simulation, quantum simulation. And the last thing I will say before questions is that it also gives us, because we have very good control over the parameters in our system, we also have a very good control, because we can also tune the parameters to values that we usually do not see in nature. So this is really a nice platform for really fundamental science, fundamental physics, because we can tune parameters to unnatural values, which will lead to unnatural effects that we can then try to harvest and use in technological applications. I think I'll stop here for questions, and then if you're interested, I can tell you a little bit about my research. So if there are questions, this is a good time.
Question for here? Yes,
What is the microscopic definition of resistance?
Well, it does not have one, right?
So how do you have any other, how do you calculate it?
No, that's the exact idea. Well, we can get into definitions. Essentially, you need to think about how to explain or how to express macroscopic observables, right, in terms of microscopic values. So, for example, you can define current in quantum system, for example. You can define a current operator and then calculate the expectation value of this current operator or response functions of current operators. But the question is, how does it relate to, how does it exactly relate to the electrons and atoms in your model? And this is not trivial to do. So that is the idea of a model.
Can I calculate a microscopic version of the resistance by dividing the expectation value of the voltage by the expectation value of the currents, using quantum expectation values in the numerator and the denominator?
You can do that. The question is what will be the meaning of this thing you calculate. You can always define, for example, current, you can always define as charge divided by time, right? So you can just measure if, for example, if you have a 1D system, it's very easy. You can just pick a position in your 1D system and then count how much charge moved for a given time, right? But the question is, how does it relate to what you measure in experiment? And this is not trivial.
Which models you were studying?
No, that was just an introduction to the field. But I'm not sure if I have time for like 2 minutes to talk about my research. Okay, good. So, great question. My research is actually a very simple system, which is a system of a single atom, right, with a single electron in a single ground state that interacts with photons, right, with light. And so this is a system you can really solve on a piece of paper. And the type of questions usually you ask yourselves is what happens when you have, for example, so you have an electron at the ground state, right, of this atom, and then you have an incoming photon that interacts with this electron, so it will excite some excited state, and eventually that will decay back to the ground state, and it will also emit a photon. Right? It will usually emit a single photon. So we'll have one coming in, one going out. And that will be, there will be no, for example, conversion of frequency of energy, right? But then the question is, could you see 3 photons, for example? Could you see more than 1? And the answer is that you usually don't see this in nature because what controls the interaction strength between this electron and this photon is what is called the fine structure constant. And because it is small, the probability to have multiple photons is very small. But then the idea is that if you can simulate this model, and this relates to the last point I said in the previous slide, if you can simulate this model and you can somehow control alpha, you can somehow increase alpha, then perhaps you will be able to see 3 photons or 5 photons or something else. And this is what we do. So the idea is to somehow design a system in which you increase your alpha, your interaction strength, and then you could see essentially splitting of this single photon to multiple photons. And this is what we do. So by we, I mean the experimentalists we work with that actually build these models. We only do calculations. So the idea is that you can somehow replace this atom with an artificial atom, which is, if you're familiar with qubits, this is a qubit, a two-level system. And you also confine your photons to move in a waveguide. And by carefully designing this waveguide, you can get splitting of this single photon. You can enhance this alpha to be on the order of 1. And then you will be able to see splitting of these photons. And the way to do that is to implement this using the superconducting circuits, but this is way too many details.
Something that you could have also just calculated in a computer, or do you actually need to implement it in order to know what will happen? That would be one. And the second question that I had was, so you're talking about taking one photon with one frequency and creating a few other photons with a few other frequencies. You have systems to do that. Are you speaking about doing it more efficiently?
Yeah.
Or
So the second question is just doing it with high probability, because usually you see, I mean, you have systems to do that, but they do that once every, let's say, 1,000 photons or milliphotons, something like that. This does it once per 1 photon. So that is the second question. As to the first question, this is actually a system that we were able to solve analytically. So we do not need the experiment to know that there will be decay, but it's good to have an experiment to show the decay, right? So this was a nice collaboration that showed the agreement between what they measured in the experiment and what we calculated. So yeah.
Okay. One more question.
Yeah, this shows the process of quantum simulation, but if I understand correctly, it doesn't have to do with a system with many particles, right?
Right. This is not, that's why I left it to the end. This is not an example of many particles. You can also explore many-body physics using these kinds of systems. This is actually in the plans, but this is, in some sense, a single-body effect. Yeah.
So, but if I understand correctly from your motivational part, most of the applicability comes from simulating systems with many particles. That's the presumed applicability?
So that more relates to the last point I said in the previous slide, which was that you can really see new physics. You can really see new effects by tuning your parameters to unnatural values.
Mm-hmm.
Yeah.
Okay. Thank you again, Amir.
Thank you.
Okay, thank you. And thank you for organizing this nice event, also Batsheva and Mira and Moshe. So I guess it's the last session, so thank you. So the work that I'm Oren, by the way. I've talked to most of you throughout this day, so you all know me by now. So the work that I'll be talking about today was completed with my PhD advisor, Yariv Kafri at Technion, and our collaborator, Julien Tailleur from MIT. And in this work, we have derived systematically the long-time dynamics of tracer particles in baths of active particles. So what does this all mean? So active particles are essentially particles that propel themselves by consuming energy. So it's something which is very familiar to most of us. Active particles are all around us in everyday life. Most examples are living examples, such as these E. coli bacteria. But there are also many non-living examples, such as these chemically propelled colloids, which are coated from one of their sides with a catalysator, and it ignites some reaction, which is polar. So they are self-propelled. And the nice thing is that we can describe both of these systems with the same physics, both the living and the non-living ones. Now, you would be tempted to look at this system from afar, from very large scales, and say, well, this essentially looks like a very hot gas of particles. So maybe it's essentially like a normal gas, but with a very high temperature, since these particles are incredibly motile. Of course, this is not the entire story, and the easiest way to tell that your active fluid is indeed active is throwing some asymmetrically shaped object into the fluid. What you will then observe is something called ratchet effects, which essentially means that you can extract work out of fluctuations by breaking both the spatial symmetry and time reversal symmetry. So spatial symmetry we break by inserting the object. Time reversal symmetry breaking means that we drive the system far from thermal equilibrium. So we can tell that by looking at the movie of an experimental system that it goes forward in time and not backward in time. Now, there are many examples of such ratchet effects in active media. One of the most famous experiments is this ratchet wheel in a bath of E. coli bacteria. So the bacteria rotate the wheel persistently on average in a single direction. Another related experiment, again with E. coli, we have our fluid, and there's a semi-permeable partition right in the middle. It is made of these V-shaped, funnel-shaped barriers, so it's asymmetrically shaped barriers. So we start with roughly an even concentration, and after a very long time, the concentration gets rectified. There is a much higher concentration on one side compared to the other. So of course, both of these experiments would not be possible at thermal equilibrium because both of them would violate the second law of thermodynamics. Basically, we would produce work out of thin air, out of fluctuations. But here we are out of equilibrium. These bacteria are active, so there's no such restriction. Now, these are very clean experiments that people do in the lab, but you could also do something simpler. Wait, what did I do? You could do an experiment in your home. You just need a glass, some honey, and a paper cone, and you got a fly trap. And basically, the flies would have a very easy time getting into the trap, but a very hard time to get out of the trap, even though the trap is fully open. Actually, some fishermen use similar devices to fish. So they have a conically shaped net, and then fish swim inside very easily, but they're having a very hard time swimming outside. And this is exactly the effect that I'm talking about. Basically, because of the persistence of these particles, they tend to travel for an extended period of time in the same direction. They get stuck on the concave side of the obstacle and flow freely through the convex side. That's the effect, essentially. Now, another manifestation of this effect can be obtained if we let these V-shaped obstacles move freely. In this case, I will call these tracer particles, since we can trace their motion. And then, if we control their orientation, keep it fixed, let's say using some magnetic field, then these tracer particles will now be propelled at a constant velocity by a constant force. So here we see simulations, and this is an experiment with, again, bacteria. Bacteria is sort of the favorite experimental setup, but as I said, there are many others. Here they actually tweak the magnetic field every now and then just to show that they can control the orientation of this particle, but otherwise it travels in straight lines. Now this is the average trajectory. What about fluctuations? So fluctuations is a fundamental problem in statistical mechanics. The understanding the fluctuations of tracer particles is extremely important both experimentally, since we use them all the time, as part of a technique known as microreology. It's a technique that allows people to measure macroscopic effective properties of very complex materials by looking at the motion of these tracers and studying their fluctuations. But tracer particles are also an important theoretical construct because they allow us to construct observable quantities that our theories could predict. And since the original works of Einstein, Langevin, and Smoluchowski over 100 years ago, we came a long way, and by now we understand the motion of the Brownian motion of tracer particles in baths at thermal equilibrium pretty well. This is not the case, however, for active baths, baths made of active particles. Although this problem has been studied for over 20 years now, we don't have a solid understanding, and here I just list a very partial list of important contributions, but there's like hundreds. So this is where we come in. We derive systematically the long-time dynamics of such a tracer particle in an active bath, and we do this in one dimension. So you should imagine a long, narrow channel filled by a viscous medium. Let's say something which is, on macroscopic scale, it would be something similar to honey, but microscopically it could be also water. And in the middle, there's a large passive tracer surrounded by many small active particles. And we consider both the general case of an asymmetrically shaped tracer of some arbitrary shape and the somewhat singular case of a completely symmetric tracer. And what we find is that the tracer symmetry is crucial for its long-time dynamics. The symmetry dictates the fluctuations. Not only dictates it quantitatively, the numbers, but it dictates the scaling behavior of this fluctuation, the qualitative behavior. So this is the main result. And now I would like to give you really a brief overview of the result without getting too much into technical details, but I can, of course, discuss this later. But basically what we do is we start with the microscopic equations of motion of both the active particles and the tracers. So we have the full set of many body problem of many particles. And then we derive from this using a systematic expansion, the effective equation of motion of just the tracer particle. This equation of motion has memory inside of it, built in, meaning that the tracer somehow has traces of the memory of the previous states of the system. It doesn't forget so easily as we tend to think of these particles do. So from this, we get this equation of motion, we compute the statistical moments of the motion of this tracer particle, and this is what we get. So for an asymmetrically shaped tracer, we find the following. We find a super diffusive expansion of fluctuations, of typical fluctuations. So what I mean by that is that the variance of the position of this tracer expands with time to the power 3/2. So about the average propulsive trajectory, we have a variance that grows as t to the power 3/2 in the long time limit. And this is much faster than normal diffusion, which grows only linearly with time. So this is why it's called super diffusion. Likewise, when you tow the tracer at a constant velocity and measure the friction force that is exerted on it by the active bath over time, we find this force to grow actually with time to the power 1/2. And here we see in numerics, this is just simulations of the microscopic models, and on top of them are analytical predictions without any fitting parameters, so that's nice. Now note here that the exponents are universal, so they don't depend on anything, do not depend on the details of the tracer or the bath. They are a statistical property of the way that active particles behave on long timescales. This is usually what we look for when we look for universal behaviors. The coefficients here do depend on the details, and we have an exact analytical formula for these that we've derived. The important bit here is that both of these are proportional to the average force exerted on the tracer squared. Recall that I told you that on average, this is propelled by an average ratchet force. And so for a symmetric tracer, there's no ratchet effect. We lose the average force. Both of these vanish, and we lose the anomalous effect. So for symmetric tracers, we have normal diffusion and a finite friction force. So seemingly, it's all boring. This is very similar to what you would have for a tracer particle at an equilibrium bath, just a dust particle in a fluid. But still, we see some traces of activity, even in this case, and we see this when the tracer size is small compared to the average distance a particle traverses before completely forgetting its previous velocity. So you should think of these particles, they walk, they change every now and then their direction. There is some average distance that they travel before completely randomizing their velocity. If the tracer size is smaller, roughly, on the order of this, it's called the persistence length scale. If it's on the order of this length scale or below, what we obtain is negative friction. Basically, active particles move to the other side of the tracer, push from there, enhancing the tracer motion instead of resisting it. And here, again, we see analytics and the simulations on top of each other. So that's nice. So to conclude, asymmetric tracers undergo superdiffusion, experience a friction force that grows with time. We've identified those as a ratchet effect. Symmetric tracers undergo normal diffusion, just like in equilibrium, just like for textbook Brownian motion, and experience a finite friction force, which can actually be negative if the size of the tracer is too small. We have analytical expressions for everything you see here, and agreement with simulations without any fitting parameters. And the take-home message here is that the symmetry of the system controls the fluctuations. If we break the symmetry of the system, the scaling of the fluctuations change completely. What we see, sometimes in physics, we call this a shift in the dynamical exponent. So you could think of this as some exponent that sort of is representative of the dynamics of the system. There are many details which might vary from one model to the next, from one system to the next, but the exponents here are something which is more universal. So this is a shift in this so-called universality class. So with that, I would like to conclude, and thank you for your attention.
Maybe I missed it, but do you have an explanation, a physical interpretation for the superdiffusion behavior for asymmetric tracers?
Yeah, well, I don't know if it's a simple one. I do have an explanation. It has to do with recurrence of random walks. So basically, the effect here is that in low dimensions, processes which are asymptotically Brownian motion-like, which are converge asymptotically to random walks, they are recurrent, meaning that the probability for them to return to the same site is sufficiently large so that when you integrate it over time, this diverges. So this divergence is really the source of this anomalous diffusion, meaning that in high dimensions, we could say that particles might hit the tracer and never return to it with probability almost 1, asymptotically 1 when you take the thermodynamic limit. But in low dimensions, there is always a residual probability to come back to the tracer and hit it again. This means that you always have a very strong memory effect. It just turns out that this leading order contribution, which comes from the recurrence, has a symmetry dependence. It depends heavily on the symmetry of the system. When the system is completely symmetric, this contribution vanishes. That's basically you get two opposing contributions from hitting the tracer from either side. I mean, it's very probable to hit it again with the same recurrence probability, but the probability to hit it from that side cancels with the probability to hit it from the other. So it's not a very simple explanation, but that's the best that I have at the moment.
Two questions. The first regarding the universality class. Is there a differential equation you can relate it to? So it's not diffusion, it's not KPZ, for example, but is there any other differential equation that will give you this exponent?
So, okay, yeah, it's not KPZ. I mean, that's the equation of motion. That's a generalized Langevin equation. It has memory. Now, this doesn't tell you a lot about the universality class, because what I've told you is just the exponent, and this describes a very wide class of processes, not necessarily with this exponent. So this is a more subtle question which relates to how to describe the probability distributions of these tracers. There are several models. I think there is not a conclusive way for a generic process which is anomalous, which has anomalous transport like this, to relate it to a model or to a particular universality class. So for example, if I would get, for example, 3/5, I would say it's supposed to be a Levy process universality class, but it's not a proof or anything. Mm-hmm. To show that it's a Levy process, I need to show that its distribution converges to a Levy process. Mm-hmm. So it's a more subtle issue. That's
okay. Thanks. And the second question, did you try to look into discrete symmetries? So if it's a sphere, so it's fully symmetric, but then what would happen if it were like a plus or something?
So here we're at 1 dimension, so there's only parity. Okay. So it's discrete. But in 2 dimensions and above, ratchet effects occurs once you break any spatial symmetry, parity or rotations, whatever. You break any spatial symmetry, you get ratchets. So I would expect this to extend.
So you expect you will get the same exponent no matter how you break the symmetry?
I mean, the exponent should depend on dimension. I have a guess for what it would be. Basically, there should be a log in 2 dimensions, as is always the case with 2D. But I mean, we haven't derived this yet systematically.
Thanks.
Okay, thank you very much.
אז אני שיר, אני סטודנט לדוקטורט בטכניון תחת ההנחיה של פרופסור עידית קידר, והיום אני רוצה לדבר איתכם על הביזנטים Among Us, או בשם היותר פורמלית של התזה שלי, Distributed Services Under Attack. אז בואו נתחיל מלהבין מה זה Distributed Services. ונעשה את זה על ידי 2 דוגמאות שכולנו כנראה מכירים. הראשונה היא מה שנקרא הענן. עכשיו, גם אם הענן לא אומר לכם שום דבר, סביר מאוד להניח שלפחות פעם אחת היום כולכם ניגשתם לענן. כן, סליחה, אני אוהבת להרצות בטיולים. אז מה זה הענן בעצם? זה אוסף של שירותים, שהם כולם מבוססי ענן, למשל ג'ימל, למשל דרופבוקס, שמה שהם עושים בעצם נותנים לנו מה שנקרא storage on demand. כלומר, אני יכולה לגשת לתיבת מהטלפון שלי עכשיו, לאימייל שלי, וזה נראה כאילו כל האימיילים שלי נמצאים על המחשב. או אני יכולה לעבור עם הפרופסורית שלי על ריוויו שקיבלנו לכנס באמצעות share doc, יכול להיות שהיא בכלל תהיה בארצות הברית באותו זמן, אבל שנינו נוכל לראות את העדכונים בזמן אמת. כלומר, יהיה לנו איזושהי חוויה שהיא seamless לקליינט של זיכרון לוקאלי. בפועל אין באמת איזשהו ענן קסום כזה שמחזיק את כל המידע, אלא מה שיש זה את אותם שרתים שמאחוריו. אז זה הדמויות הצבעוניות שלנו, במקרה הזה יש לנו n שרתים, אז n שווה 5, והשרתים האלה הם א', decentralized, כלומר אין לי איזשהו מחשב מרכזי, אלא כמה ישויות נפרדות שמשתפות פעולה בשביל לתת לי את אותה חוויה של אחסון, והם יכולים להיות distributed, זאת אומרת יכול להיות שהדמות הצהובה היא נמצאת בישראל, הדמות הירוקה היא נמצאת בקליפורניה, והם איכשהו כולם צריכים לשתף פעולה, כדי להסכים על מה מצב המערכת, על מה כתוב באימייל ששלחתי אתמול בערב. איך הם עושים את זה? אז אם מחשב רגיל מרכזי אחד יש לו איזשהו פרוטוקול, איזשהו אלגוריתם, כמו מתכון שהוא עוקב אחרי הצעדים שלו, עכשיו יש לנו גם כל אחד מהדמויות האלה עוקב אחרי איזשהו מתכון, אבל המתכון הזה מערב משתתפים נוספים. ואיך אנחנו מתקשרים בינינו על ידי שליחת הודעות. אז למשל, במקום שתהיה לי סתם איזושהי לולאת ווייל כזה, תספור עוד 10 ואז תעשה 1, 2, 3, אני יכולה להגיד, טוב, אני עכשיו מחכה עד שאני אקבל 5 הודעות, נקרא את התוכן שלהן, ואז אני אפעל בהתאם אליהן. דוגמה שנייה, שכנראה שלא כולכם משתמשים בה, אבל היא גם צוברת תאוצה ועמוסה להרבה מאוד מחקר בתחום שלנו בשנים האחרונות, היא בלוקצ'יין. אז מה זה בלוקצ'יין בעצם? אופס. בלוקצ'יין זה איזשהו ניסיון, זה טכנולוגיה, שהמטרה שלה היא לייצר לנו מה שנקרא איזושהי מערכת אמינה לניהול נכסים או לניהול מטבעות, אולי אפשר להגיד זה יהיה הכי קל לדמיין. איך שאנחנו מכירים את זה כיום, אם אני רוצה להעביר 5 שקלים לאורן, עם כרטיס אשראי, לא מזומן, הכל הולך לעבור דרך הבנק. יש לי את הישות המרכזית הזאת. מה הבעיה בפתרון הזה? שכולנו משתמשים בו היום. אבל מה הבעיה איתו? מה זה? עמלות, נכון. מה עוד? מה זה? ריכוזיות, נכון? אם מחר הפקידת בנק שלי מתפטרת, ונגיד שהיא הפקידה היחידה באותו סניף, אין לי מי שינהל לי את החשבון. אוקיי? יש לנו איזשהו גוף יחיד אחד, שברגע שהוא עלה באש, לא יודעת מה, נשרף הסניף, הלך הכסף שלי. וגם עמלות, אוקיי? זה גם, אני לא אדבר על זה, אבל עמלות זה גם נכון. אם אני רוצה עכשיו להעביר כסף למשפחה שלי בארצות הברית, ואני ממדינה, אפילו מדינת עולם שלישית לא יודעת מה, באפריקה, או ישראל, זה הולך לעלות לי הרבה מאוד כסף להעביר דרך הבנק. אז גם את זה בלוקצ'יין באמת פותר. אז מה עושים? במקום לקחת את הבנק הזה, שוב, נחליף אותו באיזה סט של תהליכים, שהם הולכים ביניהם להסכים על מה מצב המערכת. כלומר, גם פה יש לנו איזושהי בעיית סינכרון. איך אנחנו מסכימים? מה הבלנס של כל אחד בחשבון הבנק? כמה יש לי בעובר ושב? למשל, אם לדמות הנחמדה, אני לא בטוחה אם זה גבר או אישה, יש 10 ביטקוין, זה לא שרק הוא יודע את זה, וזה לא שיש בנק יחיד שיודע את זה. יש פה 5, אבל עשרות או מאות או אלפים של שרתים, שכולם הסכימו ביחד שזה כמה כסף שיש לו בבנק. אז בעיקרון שתי הדוגמאות שנתתי לכם הן אמנם אפליקציות שונות, אבל הן די דומות, הן פותרות במהותן את אותה בעיה. איך אנחנו מסתנכרנים על מצב המערכת, איך אנחנו מסכימים מה כתוב באימייל או מה כתוב בחשבון בנק שלי. נכון, אנחנו צריכים כולם להסכים. אז למה בכלל צריך להמשיך לחקור את זה? כלומר, Cloud Computing זה נושא שנחקר כבר לפני עשרות שנים, למה עדיין יש מחקר בתחום הזה היום? אז יש 2 הבדלים מרכזיים. אחד, הוא שהסקייל הרבה יותר גדול. אוקיי? אם מקודם היה לי שירות של ג'ימייל, אז יש לי את השרתים של גוגל, אני לא בטוחה, אני מנחשת שיש להם בין מאות לאלפים של שרתים מפוזרים ברחבי העולם, אז יש פתרונות שיתאימו להם. עכשיו כשאני מדברת על ביטקוין למשל, יש לי כבר מיליוני משתמשים, אוקיי? יש לי הרבה יותר דמויות שצריכות לשחק במשחק הזה ביחד. אז פתרון ל1,000 שרתים ופתרון ל1,000,000 שרתים הוא לא אותו דבר. והדבר היותר מעניין לדעתי, זה האם אנחנו יכולים לסמוך על השרתים שלנו. כלומר גוגל, שוב, סתם נתפסתי להם כדוגמה, קחו את החברה האהובה עליכם. יש להם את השרתים שלהם. אומרים, טוב, יכול להיות באמת ששרת פעם ייפול, אבל זה שרתים הם משלנו, אוקיי? הם הולכים לעשות מה שאני אומר להם לעשות. עכשיו שאנחנו משחקים בביטקוין למשל, אז אולי יש שרתים ספציפיים שיש להם אינסנטיב משלהם. נכון, אולי אני אכניס דווקא את הטרנזקציה הזאת, כי זה ככה אני אקבל יותר כסף בעצמי בתור קורא. ויותר מזה, אולי יש שרתים שכל המטרה שלהם בחיים זה להרוס לכל היתר, אוקיי? אז למשל, חשבתי על זה היום בבוקר, לא יודעת אם אתם מודעים, הייתה פריצה לשרתים של הטכניון. אולי יש אנשים שהאינטרס שלהם זה רק לחבל לנו, אוקיי? אז זה בדיוק מה שהמחקר שלי בא לענות עליו. איך אנחנו בונים מערכות מבוזרות שמצד אחד תומכות בהרבה משתמשים, הן מאוד סקלביליות, ומצד שני הן לוקחות בחשבון שיש אנשים שרק המטרה שלהם זה להזיק. כלומר, כשאני מתכננת איזשהו אלגוריתם, אני אומרת, טוב, אני עכשיו שנייה נכנסת לכובע של האיש הרע, איך אני דופקת אותו, שהוא לא יעבוד, ואז מנסה דווקא לתקן. אז עכשיו דוגמה ספציפית על המחקר שלי, יש, זה לא שלי, הלוואי, חברה שקוראים לה אלגורנד, זה אחת מחברות הבלוקצ'יין, אני חושבת מ-2018, והיא שווה $100 מיליון. מה שהיא עשתה, כל יתר החברות זה גם חברות בלוקצ'יין למיניהן, הם נתנו את הפתרון הראשון, איך אנחנו יורדים מפתרון, שוב, של איזושהי מערכת asset transfer כזאת, ששולחים בה n בריבוע הודעות, או סדר גודל של n בריבוע הודעות, למשהו שהוא יותר מתקרב ללינארי. אוקיי, אז פה שוב אם אנחנו חושבים על הענן ואומרים, טוב, ב100 שרתים ההבדל בין n בריבוע ל-n הוא לא כזה גדול, אבל כשאני ב-n שווה מיליון, אז זה כבר נהיה משהו שהוא מפיזיבילי ללא פיזיבילי, בכיוון השני. איך הם עושים את זה? אז ממש על קצה המזלג, אם תחשבו על, לא יודעת, על שיטות ממשל, נגיד דמוקרטיה. דמוקרטיה ישירה, אנחנו לא יכולים לנהל פה, נכון? לא יכול להיות שכל, אני לא יודעת כמה, אנחנו באוכלוסייה היום 9 מיליון האנשים יתנקזו לחדר אחד ויקבלו החלטות ביחד. אז הם עושים דבר דומה. הם אומרים, טוב, במקום שכל המיליונים פה יריצו ביחד את הפרוטוקול, אנחנו נדגום איזושהי תת קבוצה, ורק הם יריצו בשבילנו את מה שהם צריכים לעשות. אז עכשיו לא מיליונים של אנשים צריכים לשלוח הודעות אחד לשני, אלא רק 4 במקרה הזה. מה החיסרון שלהם, של הפתרון שלהם? הוא מסתמך על משהו שנקרא סינכרוניות. כלומר, הם מניחים שלכל 4 משתתפים, או לכל המיליונים, כי אנחנו לא יודעים מי ייבחר לוועידה, יש איזשהו שעון. ואז איך הפרוטוקול נראה? אני אומרת, טוב, אני מחכה 5 דקות, מסתכלת, הנה ההודעות שקיבלתי, זה מה שאני עושה. מחכה עוד 5 דקות, וככה בעצם הפרוטוקול שלי מתקדם. מה שהמחקר שלי עושה, זה בדיוק להעיף את השעון הזה. אוקיי? אם תחשבו על המציאות שאנחנו חיים בה, על מערכות, איך שהן פועלות באמת, כשאני עכשיו שולחת הודעה במחשב מנקודה א' לנקודה ב', אין באמת איזשהו מספר קסם כזה שאומר, טוב, תוך 5 דקות זה בטוח יגיע. בהסתברות גבוהה הוא כנראה יגיע, כן, באמת הרבה פתרונות הם בגלל זה לא מניחים הסינכרונית. אבל אם ממש רוצים להיצמד לעובדות, אין כזה מספר. אז הפתרון שלי הוא שוב, הוא יורד מה-n בריבוע הזה לסדר גודל שהוא יותר קרוב ל-n, ומסיר את ההנחה של הסינכרוניות, כלומר אני עוברת לעולם אסינכרוני. איך אני עושה את זה? אני כבר אשמח לספר לכם אחרי ההפסקה, מפאת חוסר זמן.
Attend this conference physically, but I'm really happy to share with you the work that I'm doing at Professor Ben's lab at the School of Pharmacy at the Hebrew University. So my research is focused on tumor on the chip, a physiological mimicking method for personalized therapy in cancer. So there's a great need to recapitulate different types of tissues, whether if it's for studying the toxicity of drugs, penetration into the blood-brain barrier and efficacy, or in general, studying the mechanism and biology of different disease states. For example, my research is focused on developing an ex vivo tumor model. Therefore, it's important to take into consideration the different cells and components in the tumor and its microenvironment, since they strongly influence the development and progression of the tumor, as well as its resistance to treatment. It's also important to consider the quick spatial growth of the solid tumor, which leads to a dynamic metabolic demand and increased hypoxia. Showing this diffusion gradient is critical in cancer research models since it strongly affects the cancer aggressiveness. Two-dimensional cell cultures and animal models currently serve as the major methods for testing tissue responsiveness to drugs, even though they have many known limitations of not accurately mimicking the human physiology. 3D cultures, on the other hand, have a significant advantage over the other systems since they better mimic drug resistance and spatial growth, similar to what's observed in vivo. Therefore, in recent years, research regarding 3D models has greatly evolved. There are several 3D culture models used in research. In our lab, we worked with pretty much all of these different models. I will now focus on spheroids and organ-on-chips since they enable high throughput and versatility. We can see on the left a cross-section of a solid tumor, and next to it is an example of glioblastoma spheroids. We can see the spheroids are cell aggregates with the physiological spatial growth and metabolic activity similar to tumors. When grown in a chip under continuous flow, there is better mimicking of the tumor by providing chemical concentration gradients, shear stress, and better gene expression profile. In other words, combined, spheroids grown in the chip under continuous flow can provide a good model for mimicking solid tumors. However, even though there are many advantages to using microfluidic chips for research, the gold standard for preparing chips is soft lithography, which is complicated, and as you can see, requires many steps for fabrication, requires specific skills, and the manufacturing time is long until we can actually obtain a PDMS chip that is ready for use. On the left, we can see that these steps in the fabrication process is what causes the fabrication to take several weeks and be quite expensive. An alternative method for fabrication is 3D printing. Using 3D printing for the mold fabrication step enables us to create PDMS microfluidic chips within a max of 2 days in a simple and cost-effective way. The main reasons for the fabricating chip using PDMS, the main reason for people and in research that they like to use PDMS chips, which is the polydimethylsiloxane, is because the polymer is cheap, biocompatible, and transparent. However, PDMS has many drawbacks. Therefore, we worked on circumventing the need for PDMS and were able to fabricate chips from plastic polymers, making the fabrication process even faster. It takes just a few hours. We were able to achieve chips that were biocompatible, transparent, which enables microscope imaging, high-resolution printing of small features, and the process of fabrication from beginning to end only takes a few hours. So going back to my project, first I designed the microfluidic chip according to the features I wanted to have using a 3D designing software. The design is sent to the printer, which prints the chip within a few hours. Once I receive a biopsy specimen taken from an individual cancer patient, I use various enzymes to disintegrate the tissue into single cells, which are then rebuilt into multiple spheres. In this way, these spheres become a personal cancer-mimicking model for each patient, since they contain the unique DNA of each patient and his cancer cells together with his microenvironment. The patient-derived spheres are grown in the microfluidic chips for several days while different drugs are tested on them and the results are analyzed. Here we can see a few examples of chips which are able to sustain spheroid growth for long periods of time under continuous flow and treatment with different drug gradients. On the right, we can see an example of glioblastoma spheroids that were grown in a microfluidic chip for 6 days and treated with doxorubicin 500 micromolar and imaged using a confocal microscope. We can see that we were able to obtain high-resolution images of the inner layers, which is important for many reasons. Here specifically, we were able to learn to what extent the drug was able to penetrate into the deep layers of the spheroids. In this last slide, I will quickly show you a very interesting experiment we conducted in collaboration with Dr. Shai Rosenberg, a neuro-oncologist and cancer genomics expert. This case involved a glioblastoma patient who tried many different lines of treatment, but was resistant to all of them and was eventually moved to a hospice. I received her biopsy and degraded it into single cells and prepared from them spheroids. We performed genomic analysis of the original tumor and spheroids. On the right, we can see a Venn diagram, which shows the preservation of the genomic landscape. What's interesting is that the mutation EGFR, P790M, appeared independently in 7 out of 11 spheroids, but did not appear in the original tumor. This is surprising because there is no information about this mutation in the literature regarding glioblastoma. This mutation appears in lung cancer in 50% of the unresponsive patients, which grants resistance to EGFR inhibitors. There is a drug that was specifically designed for this mutation, a third-generation EGFR inhibitor, osimertinib. Meaning, this finding yielded a new treatment idea for this patient and showed the importance of personalized medicine. So, thank you all for listening, and I would love to answer any questions you might have. You can just search my name under Liana Steinberg. Thank you so much.